Fantastic. Thanks so much for the kind introduction, Mariel, and welcome everyone. I'm joining here today from Treaty 6 territory in the homeland of the Métis in Saskatchewan, Canada. And I really like that framing of starting it out with something joyful. And so I'm providing you here with a nice little riverscape of the South Saskatchewan River in Saskatchewan in a fall of, I believe, 2 years ago, which is where a lot of my research is actually situated and framed. So today I won't be talking all that much about the Salish Sea per se, but work we did here in the province. And hopefully those are some learnings that you can take away and actually apply somewhere else as well. So I want to dive right in. Innovation in ecotoxicology is how I framed this and titled this, Integrating Next Generation Chemical and Biological Tools. And this is really something that I've been working on together with my team here for many years. And I'm really excited to be talking about this to some extent here today, at least what I can present in a short period of time. Okay. So quick overview of what we want to look at today. I first want to start this off with a bit of a view at the status quo in environmental monitoring and risk assessment. Big question that really looms nowadays is, are those approaches that we've been using still relevant today? There's a changing chemical landscape. We have new tools at our fingertips. So there's really this need to try and reimagine how we actually do risk assessment and environmental monitoring. So I want to give you some ideas on that, just to set the frame. And then I want to highlight two case studies. The first one is situated here in Saskatchewan, Canada, really trying to integrate chemical analysis and transcriptomics to assess contaminants in water, sediment, and fish. And then the next one is something that a lot of you folks will be very familiar with and potentially also very interested in, and that is using these modern chemical analytical tools to characterize biotransformation products of novel entities, novel chemicals, and in this particular case, 6-PPD quinone that a lot of you will be familiar with. And I must apologize in advance. My voice is going in and out a little bit today. Our province is blanketed in wildfires right now, and so there's a lot of smoke in the air. That's really rough on the vocal cords. So if my voice goes out and I have to take a little zip, please accept my apologies on that. Okay, so quick setting of the stage. Chemical pollution is really one of the greatest threats globally right now that puts the sustainability of human activities on the planet at risk. There's certainly a lot of reasons behind that, but you see some of the impacts here, both environmental and human health, that we are concerned about. Now, we have certainly to try and combat those issues, put legislation in place that mandate both the prospective as well as the retrospective risk assessment and monitoring of contamination, state of the environment, as well as potential impacts of chemicals, right? And those efforts are oftentimes based on priority lists of chemicals of concern. You see some of those up there, polyaromatic hydrocarbons, dioxin-like chemicals such as dioxins, furans, and PCBs, as well as a variety of legacy pesticides are oftentimes part of those priority lists of chemicals of concern. And so now their monitoring using various chemical analytical methods is mandated across a variety of jurisdictions. That is really only the tip of the iceberg, though, because if you look at the way our society has been using chemicals in recent years and decades, we really see that mismatch that these strategies and approaches that have been developed based on a relatively simple chemical space are based on chemical by chemical assessments are really unable at times to tackle this ever-increasing number of chemicals that we use in society. And that starts from pharmaceuticals to personal care products, household and construction chemicals, as well as plant protection products, and many, many more. All of this leads to the presence of complex mixtures of, at times, emerging chemicals in our environment. And the risks of those, in particular in a mixture, are quite poorly understood. And we really have this big gap in our knowledge of what we need and what it would take to protect the environment from potentially harmful impacts of these chemicals. Now, let's look a little bit at our chemical space versus available safety information. And This is very crude. You can look at this in many different ways, but one way to count the number of chemicals we know is to look at the Chemical Abstract Service. And that's a huge database, essentially a registry of known chemical substances. And it currently lists more than 219 million chemical substances. And that is of December 2024. About 15,000 chemicals are added to that list daily. And now, admittedly, not all of those are used in our everyday lives, but a lot of them could be or have at least been synthesized at least once in the process of research and development. Now, if we look at the number of chemicals for which we have safety information available, it's a much smaller fraction. That little green dot there is about 10,000 chemicals, a small fraction of those 219 millions for which we actually know what their environmental risks, their toxicological risks might be. Now, if you compare that to the number of chemicals that are present on priority lists of chemicals of concern, there's only a few dozen on those. And so you can really see that big mismatch between the diversity of chemicals out there and the chemicals we are presently monitoring in the environment. Now, this clearly shows that there are some limitations in our current monitoring approaches using targeted methods. We use chemical analytical tools that are really designed to find the proverbial needle in the haystack. So we try to take that entire diversity and boil it down, extract chemicals from it to then be able to analyze them on our chemical instrumentation. And that naturally limits what we're looking at. In order to keep up with that rapid development in the chemical space, we actually need methods that do the entire opposite, to essentially characterize that entire haystack all at once, all at the same time, ideally, right? And so advancements in chemical analytical techniques in recent decades have enabled us to get closer to that. It's quite aspirational. But at the same time, soft ionization techniques that essentially leave the chemicals more or less intact, as well as high resolution mass spectrometry, which really enables us to look at complex samples with very high confidence and specificity, have enabled us to really develop non-target approaches that promise just that. And now, mind you, I do not particularly like the analogy here. Please don't judge me for the firearm analogy in that, but I have yet to come up with a better analogy for this kind of comparison of benefits of non-target chemical analytical methods. So say we are attempting to learn something about this environment. Currently, it's a black box in the middle of your screen. And we want to study it using chemical analyses. Let's start with our status quo with the targeted methods. I liken this a bit to a sniper rifle. You try to zoom in on one particular point of that box, and you repeatedly look at it with your high confidence and highly specific methods, and you learn a lot about a small subset of that entire system. In this part here, that upper left corner of our black box, really doesn't tell you all that much, right? Now, if we go at this with our non-target methods, they are more like a shotgun approach. And this is when you hear about shotgun proteomics and those kinds of methods. This is exactly what they are attempting to do. We're trying to learn a little bit about different sections of this black box all at once. And by doing so, we get almost this scattershot of information across the diversity of corners of this system. You can already tell, okay, here's a couple of different colors in there, a couple of different things. If we do this repeatedly, the image gets clearer and clearer. And then all of a sudden we know, oh, this is probably a duck. And indeed, it's a mallard duck, right? And so using these non-target methods, we can learn a lot more and a lot quicker about a system, maybe not in a quantitative fashion, but in a semi-quantitative fashion, compared to doing this in a targeted manner. Now, in the chemical analytical space, when we talk about non-target methods, we oftentimes think about high-resolution mass spectrometry. In our lab, we use the machine in the bottom right there. It's called an Orbitrap, and it really offers a high mass accuracy, and by offering that, it offers us greater capabilities of conducting non-target analyses. And so this example on the bottom here, you can see with a low resolution mass spectrometer, you would confuse these actual two peaks that are hidden behind here as just one peak with a mass of 477.8 Daltons. But in reality, when we look at this, it's co-alluding on a regular resolution mass spectrometer. You can actually separate those two peaks here on your high resolution mass spectrometer as two separate chemicals. And this is really this method that has given rise to our ability to look at tens of thousands of chemicals in complex mixtures that we sample from the environment all at once. And this has really revolutionized environmental monitoring. Now, with the availability of high-resolution mass spectrometry, we get data files that are huge. So for one experiment, this could be several terabytes of data that we now need to sift through because we collect all the information. This is great if you think about it. You can archive the information somewhere, and retrospectively, another analyst could come along and say, okay, now I'm interested in this novel chemical that we just learned about, and I want to go back and look at Markus's data. They can do that. It's really powerful. But at the same time, you really need complex data processing and analytical capabilities to be able to tackle that complexity. There is some commercial software that we use. For example, this screenshot you see here from Compound Discoverer, which is offered by Thermo. But oftentimes you actually need to write custom chemoinformatics software. You need to have some coding capabilities, and that certainly also represents a bottleneck in terms of the qualification of your staff members and your team members, right? So definitely a limitation there to it. Now, on the effects side of things, then we move away from the chemical analytical space. Molecular toxicology methods have undergone a very similar trend. So similarly, back in the day, you would have gone out and taken a sample, a biological sample perhaps, and determined some biomarkers, some enzyme activities, those kinds of things in a very targeted manner. Nowadays, we can actually take samples from an organism, either from the field or in the lab, and in a very unbiased fashion, characterize broad molecular perturbations of those systems through either next-generation sequencing techniques or other high-throughput techniques, such as qPCR arrays, one of which has been developed here in the Toxicology Center, the Ecotox chip. You see one of those down there. Really, again, large data sets need specialized bioinformatics skills to analyze them, but if you do have them available to you, you can actually learn a lot about a biological system very, very quickly. And so it's a very powerful method. Now, all of this, and admittedly, I'm just scratching the surface here in the interest of time to get through this. This raises a lot of conceptual and practical questions. So, for example, what fraction of the chemical space can we actually detect using our methods? There will be some chemicals that are detectable based on soft ionization techniques and based on the mass spectrometry we are applying here, while others are not. Now, what fraction of the detectable chemical space is actually toxicologically relevant? We may assume that most of the chemicals we're able to detect have little to no toxicological relevance. And so how do we tease out the ones that do have relevance, right? So that's important. In both the chemical analytical and the effects data sets, they are more and more complex. So on the one hand side, how do we bring them together? Either through informatics tools, through novel software, or simply conceptually, right? So there are different things we're looking at. And it's important that we get the semantics as well as the syntax right in doing this. Now, in the end, if we can use these methods, either in combination or alone, can we actually use them to inform prioritization efforts? So for example, we go out and take an environmental sample, we measure 10,000 chemicals, and we want to know which maybe 5 or 10 of these 10,000 chemicals are most relevant and should be part of some of our cleanup efforts. then we really need to develop concepts, approaches, and tools to be able to do that. So I want to dive right into some case studies here. And this one is from Saskatchewan, and the next one is a bit more relevant to the global audience. But this first case study here is situated in Wascana Creek, which is near Regina, the capital city of Saskatchewan, the beautiful province that I'm in. It's not quite as smoky as it is currently in Canada. It's very heavily polluted, wastewater-dominated stream. It's made the news multiple times for the presence of a variety of chemicals, and also the fact that there is frequent algal blooms, as you can see on the bottom right picture here. It ultimately discharges into the Qu'Appelle River and then in the Assiniboine, which all the way then into Lake Winnipeg Basin it will continue. So local study, but it is connected to a larger watershed. This is all work that my former master's student, Anna Cardenas, has been doing in our lab. Really outstanding work, and you can see a picture of her not in Wascana Creek, but in the South Saskatchewan River down there. There were some recent upgrades to a wastewater treatment plant in Regina, which was intended at least to improve the wastewater quality in Wascana Creek. There is some indication that that's the case, but not perhaps to the extent one would have hoped, because there were very high concentrations of endocrine disrupting chemicals in the effluent and in Wascana Creek that are really worrisome. Information on other chemicals was unavailable at the time, so we were also trying to fill some of the gaps related to the diversity of chemicals we find in this system. Now, in terms of methods, Anna went out and collected water, sediment, and fish, in this case fathead minnows, in the spring, summer, and fall of 2021. And she did that both upstream and downstream of the outfall of that Regina Municipal Wastewater Treatment Plant. Now, she took duplicate grab samples of water and sediment, and then also 10 replicate fish at each site and each time point for chemical analysis. And then also 6 replicate passive samplers, which you can see on the right here, which we call diffusive gradients in thin films passive samplers. So they essentially are a stack of different gels and membranes in a plastic housing that really has the ability to, over time, sample chemicals from the environment, and by doing so, enabling the assessment of what we call time-weighted average concentrations, rather than those grab samples and snapshots in time, right? So you get essentially an estimation of the exposure concentration over the 2-week deployment time of these samplers, or however long you deploy them in the field. Now, we subjected all of those samples to both targeted and non-targeted chemical analysis. The non-target analysis was done using a so-called data-dependent MS2 workflow. That essentially means that at each time point of this analysis, every single scan of the mass spectrometer, it will look at the top 5 or top 10, in some cases, most abundant ions, and will then fragment them further to look at the fragmentation pattern. And that really gives you a lot of confidence in the data you are creating. Rather than looking just at the parent mass, you're also looking at the fragmentation pattern. So that's almost like a fingerprinting for your chemicals that you're looking at. Now, of those fish we sampled, we took additional fish at each site, and actually also took brain samples of these, 5 to 8 replicates, depending on the site and what was available. And we conducted Ecotox chip analyses. Now, this Ecotox chip, as I mentioned earlier, it's a qPCR array, which has upwards of 350 genes on there, which you can simultaneously screen for their increase or decrease in transcript abundance in your exposed organisms. So it's a really powerful tool situated somewhere between your targeted PCR analyses versus your non-targeted, say, RNA-seq or transcriptomics analyses, which can be quite costly. Now, all of this has been published in this paper down here. So if you're interested, have a look. I encourage you to go have a read, because I can only glance over so much information in the short available time. Okay, so let's dive right into some of the observations here. First, for the targeted analysis, we picked a list here of 7 targeted chemicals. All of these, because this was part of a larger project, are neuroactive pharmaceutical drugs. Amitriptyline, bupropion, carbamazepine, clozapine, fluoxetine, lamotrigine, and venlafaxine. And so we looked at all of those 7 in sediment, in those DGT, time-weighted average extracts, as well as in fish. And you can see down here, spring, summer, fall, those are our 3 seasons. And then in colors, you can see the upstream samples are in orange and the downstream samples are in purple. And you can instantly tell from this, especially when you look at those water concentrations in the DGTs here, that the downstream samples are always orders of magnitude greater in concentration compared to the upstream samples. In some cases, you will see that, especially in the fall sample here, that the difference is not all that pronounced, and that has to do with the fact that under some conditions in the fall, when the discharge, the flow in Wascana Creek is low, you will actually get a reversal of flow because of the high discharge of wastewater effluent into this little creek. And so you get a bit less of a pronounced difference there, simply because the upstream might be impacted by wastewater as well. You can see very similar trends in the fish here. Oftentimes, you get a higher concentration in your downstream samples compared to the upstream samples, and the same is true for the sediment samples, even though those trends are not as stark as in the water samples. That likely has also to do with the fact that, the sediment will be reflective of a history of that flow reversal under low flow conditions, and your fish may also be somewhat mobile, but in general, the trends here are very similar. Okay. Now, if you look at this a slightly different way, again, we have our fish concentrations up here. Those are the exact same that you would have seen in the earlier slide. We also calculated bioaccumulation factors just relative to water concentrations and fish, so field-based bioaccumulation factors, as well as biota sediment accumulation factors. And you can see this black line that goes throughout here marks a bioaccumulation factor of 5,000 liters per kilogram. So this really indicates potential for bioaccumulation of those compounds. And you do see that, especially for bupropion, clozapine, fluoxetine, and venlafaxine, you do see exceedances of this bioaccumulation factor of 5,000, which really does indicate that those compounds have a tendency to accumulate in your fish here relative to the ambient concentrations. Now, this is fairly easily handled because it's a list of 7 chemicals. We know exactly what we were looking at in the different matrices. And so that was quite important for us to look at, just to internally validate essentially what we're looking at with our non-target chemical analysis. So we did quite a lot of extensive filtering and QA/QC just to make sure that we do not count compounds into our analysis that would be present in either our samples or in field blanks or just background that you would find generally in a lab, because these are based on detection above a certain threshold, certainly that is, but not necessarily on quantification, right? So we're looking at this simply as a presence-absence type of analysis. So it's quite important to get that filtering and QA/QC step right. We did all of that in Thermo Compound Discoverer software, and it did reveal that there were hundreds of distinct chemicals in the DGT extracts, the sediments, and the fish across all 3 seasons. So you can look at those Venn diagrams here. Our fish in the spring, in the summer, and the fall are shown down here, and then the corresponding analyses of environmental levels up here as well. What is interesting is that only a small fraction of those compounds, you see those here in the middle, were present in all 3 environmental matrices. So in the spring, you see 9 compounds are present across water, sediment, and fish. In the summer, it's only 1, and in the fall, it's 5 of them. So that already is an important indication that only a small fraction of these compounds will actually be bioavailable and will be relevant, perhaps toxicologically, to our fathead minnows. That is all certainly assuming that that we get the dilutions and extractions, etc., right. There's all sorts of processes that could lead to biases in that assessment, so you really need to be careful when you do something like this. But it is really good first indication that we can use this as a prioritization scheme, right? Okay. Now, you can look at this diversity of chemicals using a variety of methods. Compound Discoverer will spit out the best match based on some databases that it's screening. Obviously, the improvement of these databases for screening is another very important objective that, that researchers are actively working on. This was not the key goal of this endeavor here. But suffice to say that this is quite important, because only a certain subset of your compounds that you actually detected will have a match in your database that you are screening against. So that's quite important. You will bias yourself one way or the other by using one over the other database. Now, we used the positive identifications where we know the chemical identity of those compounds that were identified using this suspect screening approach, and use them as input for the CompTox chemicals dashboard. And based on that dashboard, you can do a batch process and essentially look at what the use classes of those chemicals are. And you can see that across all the matrices and sites, pharmaceutical drugs actually dominated the chemical use class. About 48 to 79% of the chemicals that were detected belong to that use class, directly followed by personal care products with 10 to 25% of the chemical use class. And then smaller use classes were solvents, surfactants, reagents, so more industrial type chemicals, as well as biocides and pesticides. Okay, so now we know the rough identity of what we're looking at, quite a bit in terms of pharmaceuticals and personal care products, which perhaps isn't all that surprising because we're looking at a wastewater-dominated stream, after all. Okay, so I think this is quite an important internal validation to see that we get the right compounds back using our non-target analysis. Now, in terms of the Ecotox chip analysis, on the top you see for both summer and fall, where we got fathead minnows to sample. These Sankey plots indicate which biological processes and functions were perturbed. And so this is the result of our Ecotox chip analysis. In yellow, you will see log fold changes greater than 1.5, and in red, log fold changes of greater than 2. So the hotter the color, essentially, the greater the perturbation in those downstream fish versus upstream fish. And you can see that in both seasons, you get quite a bit of perturbation across different systems. And then if you break it down further, you actually see there's quite a bit in terms of endocrine disruption and metabolism, as well as some immune processes. Now, that's the one indication from our fathead minnows and their response that we have analyzed. You can also screen the chemical list from your non-target chemical analysis for their toxicity profiles. And again, this is something you can do in the CompTox chemicals dashboard. Essentially, you batch process and you look for available information on the toxicity of these compounds, including ToxCast and Tox21 databases, which are readily accessible through that. And so what you see is that between those two analyses, both are experimental effects that we determine in our exposed fathead minnows versus those predicted from the chemicals present, that there's quite a promising congruency and predictivity between those two analyses. So quite promising. Admittedly, it's early stage in this, and we definitely need to validate this in other systems and other species, other conditions. But for the time being, we are quite encouraged by this positive outcome. Now, I mentioned earlier that we can use this for prioritization purposes. And so in this particular case, we essentially derived a list of compounds that co-occurred with the biological perturbation of molecular processes in fathead minnows, and in that case are essentially predictive of those potential effects here. And again, this is not necessarily causality. This is, at this point, it's a correlation essentially between the occurrence of those compounds and effects that would be explained by the presence of those compounds. But it's really a good start. So if you look at these, there's a number of different ones in there, some of which are known culprits. Propranolol is known to cause biological effects quite a bit. Lidocaine is actually an anesthetic, which we know has biological effects. Things like clozapine and desmethyl citalopram, antipsychotics or antidepressants. Those, again, are known to perturb biological processes in brain tissue of fish. So this is a really nice finding here. And then too, and this ties into the next case study, there's one industrial chemical in here that is quite an interesting one. N,N-diphenylguanidine is actually across many of the non-target chemical analyses we do in environmental matrices, one of the most frequent detections. And this is a rubber tire-derived chemical that has been detected quite broadly across stormwater samples in particular, and oftentimes is the most abundant of all the chemicals in there. So that too indicates that perhaps this is a compound that might actually lead to some of the biological perturbations we see in fathead minnows. So definitely something we want to look at. Now, quick summary of this first case study. We were able to conduct non-target chemical analyses for water, sediment, and fish in Wascana Creek here in Saskatchewan. We're able, based on that, to prioritize 10 chemicals for further study. Those would be quite interesting to look at with more quantitative methods, but then also to conduct associated lab exposure experiments to see if those correlations actually hold true under lab conditions as well. Now, this Ecotox chip analysis did predict toxicity profiles that showed remarkable similarities with those that you would expect just from the present chemicals. And like I mentioned earlier, this study is limited in terms of scope, but the approaches and methods that we describe here have great potential for serving some of those really pressing issues in other ecosystems and watersheds. So we believe this is quite powerful and should be applied and tested in more systems. Okay, now to the second case study. And as I mentioned, this is something that a lot of you folks will be very aware of. A lot of your researchers in the area are active in this space, and our activity perhaps is a bit more peripheral to that central effort. But nonetheless, I want to give a quick overview here, probably preaching to the choir, of what the issue is. So since the 1990s or so, coho salmon in the Pacific Northwest have shown these strange symptoms that you can see in the video down there. Essentially, whenever it rained after stormwater runoff events in urban streams, you saw these gasping fish at the surface and spiraling motion, which ultimately led to death. Oftentimes, that was actually in pre-spawn fish, and so it has been dubbed this so-called urban runoff mortality syndrome, which essentially is a specific version or a specific outcome of a more general effect that we oftentimes see in salmonids and what we call pre-spawn mortality. It took quite a while until 2021 until the actual chemical culprit behind this urban runoff mortality syndrome, or HRMS, has been identified. This is the 6-ppd quinone that is now studied all across the globe by groups in every corner of the world, it seems. So this seminal study here by Zhenyu Tian out of Ecology's group has really sparked a lot of interest in research and a lot of follow-up interest. And they really applied a very, very elegant workflow, again, using high-resolution mass spectrometry to identify what compound out of out of tire wear, which has been suspected as one of the driving forces here because of the close vicinity of those HRMS events to roadways and the co-occurrence with rain events. So they applied this workflow to essentially reduce the complexity of the chemicals present in tire wear leachate using fractionation techniques down to a fraction that only had 4 compounds left in it, but was still toxic to coho salmon. So in that particular fraction, you can see this little orange speck down here. Only 4 chemicals were present, and 1 was most abundant and was later characterized as a 6-ppd quinone. Now, what is 6-ppd quinone? We actually don't add 6-ppd quinone to our rubber tires. It's an environmental transformation product of 6-ppd, which is added to tires to prevent it from becoming brittle. So you can see this here. There is a rubber sample with 6-ppd and one without. You can see that without the 6-ppd, you get a really brittle surface of the rubber, which is because now the sacrificial molecule isn't there, and oxygen, ozone in the ambient atmosphere will actually start attacking the rubber polymer itself rather than the 6-ppd. So by sacrificing itself and turning into the quinone, the 6-ppd protects the tire from becoming oxidized itself. So this was quite an important discovery, and like I mentioned, it sparked a lot of follow-up interest and a lot of subsequent research. We did quite a bit of work in our group as well, and I will only present a little subset of this work here today as it relates to high-resolution mass spectrometry. Now, throughout this research journey, it was essentially found that coho salmon are exceptionally sensitive to 6-ppd quinone. The LC50, so the median lethal concentration that kills half of organisms in a lab experiment of coho salmon, that is, was 95 nanograms per liter. And with that, 6-ppd quinone really ranks among the most toxic environmental chemicals known. There's a bunch of other chemicals that are more toxic, but those are normally either biotoxins or pesticides, which are actually intended to kill target organisms, right? So it's quite surprising to see that high level of toxicity for an environmental unintended transformation product of a chemical. Now, except for coho salmon at the time, other fish species, invertebrates, whatever was tested at the time was found to be insensitive. So at that point in time, we didn't quite know if there were any other species that were also sensitive to 6-ppd quinone. We knew that environmental concentrations of 6-ppd quinone in stormwater quite frequently exceeded those that are lethal to coho salmon, including here in Saskatoon. So Ed and Jen Yu gracefully provided us with a standard, which we then used to quantify 6-ppd quinone in stormwater runoff from Saskatoon here, where our University of Saskatchewan is located. And lo and behold, we found it at levels that exceeded those toxicity threshold for coho salmon. So we definitely asked ourselves, how widespread is this sensitivity across species of fishes? What drives potential differences in sensitivity? And is this compound relevant only to the coastal ecosystems of the Pacific Northwest, or is it more broadly relevant across the continent and perhaps across the globe? Now, first I want to start this off because it helps to situate our high-resolution mass spec efforts with some of our studies on the acute lethality of 6-ppd quinone. So we conducted a number of lab experiments to assess the acute and subchronic toxicity of 6-ppdq to a variety of fish species. And we selected them to be of commercial, cultural, and also ecological importance. Now, with subadults and juvenile life stages, we conducted 12 to 96-hour acute toxicity test, depending on how sensitive they were. Some of them had to be terminated a bit early. And then we also conducted 28 and 45-day subchronic tests with rainbow trout and lake trout. So all the way from hatch to past swim up, so that we could actually estimate the effects that 6-PPDQ might have during this important developmental stage of these two salmonids. Now, we conducted static renewal experiments, about 70% water changes every 24 hours. And while that was fairly easy to do for the acute toxicity tests, you really need to have a bunch of really dedicated grad students to do that for 28 or 45 days for rainbow trout and lake trout. And so we are really lucky to have some of those here in the Toxicology Center. And so this is all work by Katie Roberts, which I will be presenting here, who just finished her master's based on this and has just started her PhD in our lab. Now, because 6-PPD quinone has the tendency to degrade fairly quickly in water, we also determined the exposure concentrations using, again, liquid chromatography and high-resolution mass spectrometry just to be able to calculate time-weighted average concentrations. Now, this is a fairly crude summary of all the findings. I will tell you this much and not show you the information. For most of the species, there was Arctic char, brown trout, bull trout, west slope cutthroat trout, and white sturgeon. We saw no mortality whatsoever up to the highest tested concentrations. Those were really unrealistically high, greater than 13 micrograms per liter, which we essentially do not see even in stormwater samples. Maybe under some very extreme conditions, but you're getting awfully close to the water solubility there of the quinone. So we didn't go any higher than this. Also to save some of the cost during our lab exposures because the quinone is very expensive. Now, for brook trout, rainbow trout, and lake trout, we did see significant mortalities with LC50 values between 0.33 and 1 microgram per liter. And you can see some of those curves here without data points on the right, just to keep it a bit more manageable. And all of this has been published in this paper here. So if you're interested, go have a look and read in more detail. Now, if we put all of this together with the information that has now been created across the world, there's also a number of literature studies there. You can see that there's essentially two camps that fish species fall into. Either you're very sensitive with sub-microgram per liter LC50s on the left, those will be these here, or you're not sensitive at all. And so even at concentrations greater than 12.7, 13 micrograms per liter, nothing happens, at least not in the short term. You don't see those mortalities. To date, it's only salmonids that have been shown to be sensitive at those low concentrations. But otherwise, phylogeny really seems to be a poor predictor of sensitivity. So you can look at some of those things here. Oncorhynchus species, rainbow trout is sensitive. Other Oncorhynchus species over here are not sensitive, right? Lake trout, Salvelinus, you get a sensitive species here, same with the brook trout, but then your Arctic char is not sensitive at all. So quite interesting to see those stark differences, even in closely related species. Now, how can we explain those differences? And this was the big question that drove a lot of this work. And so we set out to try and answer this. From the start, and there were a couple of indications, we suspected that there were some differences in the ability of species to biotransform 6-PPD quinone as a potential driver of species differences in sensitivity. Most notably, the insensitive species in lab experiments showed a more rapid drop in the water concentrations of the quinone compared to the sensitive species. So to us, that indicated, oh, perhaps there's some biotransformation going on here. And now the big question was, is it detoxification? Is it bioactivation? We didn't quite know at the time, and perhaps we don't quite know yet today. There's still, I'd say the jury is out what might be the case. But we set out to collect gallbladder bile samples as well as whole body samples of those subadults or juveniles, depending on their size and age. We also conducted incubations with cell lines and hepatocytes from rainbow trout to identify some of the possible enzymes. And again, and this is where it all comes back, we conducted a data-dependent MS2 non-target workflow using our Orbitraps to shed some light on this mystery here. Now, taken together from all of those studies, we came up with this biotransformation pathway. So essentially, your 6-ppd quinone will be hydroxylated by cytochromes P450, either on that phenyl ring here, we call this TPOH2, or on this alkyl side chain here, and we call this TPOH1. We have actually verified those two biotransformation products structurally using neat standards, which we got customly synthesized by Cayman Chemicals. So really important validation work that has been made possible by a collaboration with this company. We also detected associated glucuronides. So you can see those here. So either this hydroxy over here will be conjugated with glucuronic acid to yield this biotransformation product, or that phenyl ring hydroxylation will do the same over here. You also see some sulfates, which are likely generated by sulfotransferases. And then we also sometimes see that the biotransformation products are further hydroxylated to yield double hydroxylated compounds, which can then be further either singly or doubly conjugated with glucuronic acid. What was quite interesting to observe is that TPOH2, so this phenyl ring hydroxylation, was most dominant, usually about 2/3 of the abundance of biotransformation products, and was also present in all the species. Why this TPOH1, the side chain hydroxylation, we could only detect in lake trout and in rainbow trout, so two sensitive species, but not in brown trout, a sensitive species. So that could be a potential indication that this TPOH1 is an important driver of toxicity also. We don't quite know yet. Now, we also looked at cell lines, as I mentioned, and there were some differences between cell lines. In gill cells, we actually saw that only the parent compound was present and there was no hydroxylation that occurred, while in liver cells, we got quite abundant hydroxylation reactions in both media that were used for cell culture as well as the inside of the cells. This was also accompanied, interestingly, by some effects that we saw indicating interferences with the mitochondrial respiration, which only occurred in the gill cells. So the one biological metrics here that did not show biotransformation, but it was not observed in our liver cells. So again, we thought, perhaps this indicates that it's actually detoxification rather than bioactivation. And all of that is published here in this paper if you're interested in having a look. Now, we did a follow-up study on this and co-exposed or pre-exposed our cells, and this was just the liver cells in this case, with known inhibitors of a variety of biotransformation products. Again, you can see here this green color, most abundant TPOH2, less abundant TPOH1. There's also another not very well characterized at this point transformation product that we don't quite know the structure of yet, which is also present. And you can see that, especially if you look over to the right here, if you look at your negative control, obviously there's no biotransformation products present as you would hope. If you just incubate with 6-ppd quinone, you get a fairly strong abundance of biotransformation products. You inhibit CYP1A using alpha-naphthoflavone, and you get an almost full inhibition of these biotransformation products. And now if you induce CYP1A using co-exposure or pre-exposure in this case with beta-naphthoflavone, you get an increase in this biotransformation. So that all together indicates that CYP1A is likely one of those drivers of that first initial hydroxylation reaction. So quite important outcome. And again, if you're interested, and there's another paper on this, which you can read more on our methods and findings. Now, we also wanted to look at differences across fish species. And so in this particular case, we looked at gallbladder bile. At that point, we hadn't yet obtained the standards. So this is all semi-quantitative. You can see it's essentially in comparative peak areas here. And we didn't have the differences between the phenyl and the side chain hydroxylation figured out yet. So this is based on overall hydroxy 6-ppd-quinone and the associated glucuronides. But you can see that with decreasing sensitivity, so here from the coho salmon all the way to your West Slope cutthroat trout, you get a decrease in sensitivity. And with that decrease in sensitivity, you get an increase in the abundance of biotransformation products, both the hydroxylated one and the glucuronidated one. And so overall, that might suggest that sensitive species cannot effectively detoxify 6-ppd-quinone. Again, there's a bunch of confounding factors in this study, but certainly a strong indication that something might be going on here. And all of that is published here in this particular paper if you want to have a look. Now, in conclusion, for this case study, we could observe differences in the ability of various species of fish to biotransform 6-ppd-quinone. It's likely a contributing factor to those species differences in sensitivity. Jury is still out on whether these are differences in detoxification or bioactivation. And so in order to hopefully answer this fairly quickly, we are currently in the process of conducting acute and subchronic toxicity tests exposing early life stage and juvenile rainbow trout to both the 6-ppd-quinone as well as both transformation products. So those are ongoing. And hopefully we'll be able to answer those remaining questions, which will be quite important to inform the risk assessment of this important chemical. Now, overall, and this is my last slide, I just want to sum this up a little bit. I believe I was able to show you that those novel non-target chemical analytical tools and methods based on high-resolution mass spec are really quite powerful in bringing together mechanistic toxicology efforts, but also improving environmental monitoring efforts. They really have the potential to revolutionize what we're currently doing. We're really early days conceptually to try and link those. And so there's really still a big research need to go deeper and to really repeat some of those analyses and assessments in other systems, in other watersheds, other species, etc., just to see if we can indeed use this reliably for prioritization efforts. Hopefully, we can actually offer an approach that helps guide decision makers and risk assessors in developing refined monitoring efforts that really have the ability to tackle this vast chemical complexity. And presently, I would suggest that these methods are still subject to ongoing research and development, and more work is needed before they can be broadly applied, but they certainly hold a lot of potential. And with that, I would like to thank all of the collaborators, all of the funders that have made this work possible. I'm certainly only, one person in this larger team that is able to present here. And I want to thank all of you for your attention, and I look forward to a nice and vivid Q&A.
Wonderful. Thank you, Markus. We welcome folks to raise their hand to ask questions, but in the meantime, we've got a few in the chat. In particular, there was a question about for that first case study. That's why CLO was one of the drugs where fish concentrations were higher upstream than downstream. And if that can just be explained by the flow reversal or not, given that some of the other drugs didn't show a similar pattern.
Oh, that's a great question. Thank you for that. Yeah, I mean, this is certainly one potential explanation of what might have been going on there. I think if I couldn't summarize all the data we've collected. There's also quite a bit of variability in terms of physical chemical properties that we measured upstream versus downstream, and then also later on in the receiving Capel River. Where we think that some of those compounds that are actually ionizable may respond in different ways compared to some of the other chemicals. I didn't really want to dive too deep into that. We did some correlation analysis in the paper, but I would suggest it's fairly preliminary. So I think it's quite important if we think about this chemical space that we are trying to tackle now, that we actually look at some of those behaviors, in particular the ionizable compounds, because a lot of the risk assessment strategies we are implying are based on things we've learned about neutral organics, right? And sometimes very, very recalcitrant chemistry, such as chlorinated compounds. And so I think more needs to be done, but definitely a good point there. I think there's more to be teased apart.
Appreciate that additional context, both on this particular example, but also on how that touches on things more broadly. I think you addressed the other question about other species nicely already, but anything in terms of just kind of sublethal impacts and how to think about lethal versus sublethal with these tools that you want to reflect on?
Yeah, no, thanks for that. And I see in the chat the bull trout. Yes, we're yet to publish this this study. We've actually done a couple of studies in species that showed to be insensitive. Bull trout was one of them. Copper red horse is another endangered species that some colleagues of ours at McGill have tested, and we're bundling those together because both of them are important endangered species here in Canada, at least. And so, yeah, for the bull trout, there's actually quite a bit of follow-up work going on right now with one of my collaborators, Steve Wiseman at the University of Lethbridge. They have been able, together with the Department of Fisheries and Oceans, to spawn bull trout and to look at early life stages of those. And they did co-exposure experiments essentially with temperature stress and 6-PPD-quinone, and I believe also with phenanthrene, another quite abundant compound in stormwater. And so also having cardiovascular effects, I think this might tease apart some of those potential combination effects we are interested in. And so that study will look at some more sublethal effects versus just the lethality, because I fully agree with you. There's yeah, there's still more to be learned about this compound rather than whether or not it kills fish.
Yeah, appreciate that. I'm just going to make a quick plug for on July 8th. We will have our next roundtable, which will be about assessing and mitigating climate change impacts on Pacific oysters. So I hope that folks can join. We'll send that out in the link, and I've dropped that in. But I'm going to go ahead and Wei, if you want to ask a question, you should be able to unmute now.
Hey, Professor Markus. Glad to see your great presentation. It's really informative for all your results and your methodologies. I have a question about your methodology during your bridging, your identification, and the ecotoxicity evaluations. The question is like at the very initial stage, how did you like bridging the novel molecules you identified, whether it is undergoing some biotic or abiotic transformations? How did you bridge these molecules with their observable toxicities? Yeah.
Okay, so if I get you correctly, are you asking about the 6-PPD-quinone transformation products or in case study 1?
I think it's in both of the studies. Yes, you have
Okay.
identified a lot of the novel TPs, but at the very first stage, you don't know which one is more toxic, which one demonstrates more like eco threatens to the surrounding environment, right? So how did you do at the very first stage to like, yeah.
Right. So in our first case study, what we ended up doing, and I might not have explained this in sufficient detail, we used the positive confirmations or detections of our compounds. Again, this was done with Compound Discoverer, so fairly conservative because the essentially we just included in this compounds that we were able to find in previously published chemical databases, right? So anything that's not in those databases, you will not be able to see. So that's a first important caveat and step. The second one essentially is that now having a compound in a database, being able to chemically ID it with a certain level of confidence based on the MS2 data, so essentially your chemical fingerprint, doesn't really tell you anything about the toxicity of this yet, right? You could now go ahead and use some predictive tools You can also do that for the compounds you have not positively ID'd to predict the predict the toxicity based on physicochemical properties. That's a possibility, but I'm not sure how high our confidence would be in this at this point in time. But with those ones that were positively ID'd, we essentially went into the US EPA CompTox dashboard and screened those compounds for available toxicity information according to ToxCast Tox21 tests, and then essentially associated those with those potential toxicities, right? Again, a fairly crude method of doing that. Essentially, we can't know about toxicity of chemicals we don't know about and that haven't been tested.
Yeah.
So again, you go down a certain rabbit hole of biases essentially in all the available information that is out there. And so I think you raise an important question, is that it's quite important to make sure that those databases are filled with the right information that helps us understand what's going on in the environment. So I think it's chemists, biologists, everyone needs to work together in order to fill in those gaps.
Yes, thank you so much for your answer. This is exactly what I'm like to ask as a follow-up questions is like, how do you think the power force, how do you think the results from these in silico predictive models like the US EPA COSA models? Because I saw a lot of scholars, they were not really convinced about the predictive toxicity results. So even though for the novel identified, for example, like the biotransformation products of 6-vitiquinones in the fishes, like the hydroxidized 6-vitiquinones, we do not have the standards as a very initial stage, but we don't want to have a very preliminary toxicity evaluation for them. So we give the predictive toxicity, but I saw many of the scholars, they do not buy this toxicity data. So I want maybe listen for about your opinion, how convincing of this predicted data, how it could be like applied for multiple disciplinaries, or it was very constrained that we still need some further in vivo or in virtual toxicity evaluations. Yeah. Thank you.
No, that's excellent. I think you raise an important point. It's certainly as input into a screening effort and into prioritizing what to look at next. I'm very confident in those methods. You got to start somewhere, and you can't do it all. So we need to use some of those predictive tools in order to inform our next steps. I believe that's a very valid use of them, right? Now, would you trust your life with that in an environmental risk assessment or a safety assessment of a compound? It's maybe a different question and a different different outcome or different context in which to use them. So I really see them as sort of hypothesis generating machines in order to then experimentally validate the findings or predictions further down the road. Maybe not as the final input into risk assessment, but if you look at prospective risk assessors, maybe you will find someone who disagrees with that. Anyway, those are my thoughts.
Thank you, Markus, and thank you for the great questions, Bai. I know we're a couple minutes over, so I think Emily and Heather, I see your questions in the chat, but if you want to maybe follow up over email with those, I just want to respect everybody's time. Thank you very much, Markus, for the insightful reflections on where technology is evolving and how that plays into research and considerations around risk. Really appreciate it and hope everybody enjoys the rest of their afternoon.
Thanks so much. Take care.
Bye.